During this lecture, we'll discuss about the receptors and their functions. So we'll be discussing different types of receptors, their mechanisms of action, receptor modulation, and regulation. So why do we need these receptors? As you know, many bodily functions require cell-to-cell or intracellular communication, such as homeostatic mechanisms. So these cellular communications through chemical messengers occur by binding of the messenger to specific target proteins known as receptors. This specificity of the receptor to its ligand is very important because chemical messengers such as hormones, drugs, and neurotransmitters cannot bind to any receptor to bring about their intended action. The specificity is an important feature because it will allow to bring about the intended action on intended cells without affecting the activity of other cells. For example, adrenaline is secreted during stress response to divert more blood flow to active muscles and less blood flow to organs not needed for rapid motor activity, such as GIT system. This selective action is only possible because of the specificity of receptor-ligand action. So all these receptors are protein molecules, and they have a specific site for binding with its ligand. We have two categories of receptors based on their location. One is cell membrane receptors that have a domain exposed to the extracellular environment, so that the messengers can bind with them outside the cell. The other one is the intracellular receptors that are located within the cell. We'll talk about cell membrane receptors first. They are protein molecules that span the cell membrane with an extracellular domain that binds with the chemical messenger, a transmembrane domain, and an intracellular domain which is coupled with the second messenger to transmit the signal inside the cell. These cell membrane receptors usually bind with hydrophilic messengers that cannot cross the cell membrane freely. Examples are polypeptide hormones such as TSH, ACTH, and LH. The mechanism of signaling of cell membrane receptors include binding of extracellular domain with the first messenger. This causes a conformational change to the cytosolic domain of the receptor, which is coupled with the non-protein second messenger, such as cyclic AMP. As signaling via second messenger is faster, they produce a rapid cellular response. There are four types of cell membrane receptors. The receptors functioning as channels open or closes upon binding with the specific ligand to allow transportation of ions through the channel. One example is nicotinic receptors that allow influx of calcium and efflux of sodium upon binding with acetylcholine. Then we have G protein coupled receptors that open an ion channel or alter an enzyme activity. A good example for this is adrenergic receptors. Upon binding with adrenaline, alpha-1 receptors lead to opening of calcium channels in smooth muscles, and beta receptors, upon binding with adrenaline, activates adenyl cyclase in the heart muscle. Next, we have receptors coupled with enzymes such as tyrosine kinase, which activate the coupled enzyme upon binding with the messenger. Lastly, we have receptors coupled with cytoskeleton, which include changes in the organization of the cytoskeleton upon binding with the its specific messenger. So this picture illustrates how a G protein coupled receptor will undergo conformational changes upon binding with its ligand, leading to production of the second messenger, which is the cyclic AMP. Next, we'll look at the intracellular receptors. There are two types, again based on the location as cytosolic and nuclear. They bind with lipophilic messengers that freely cross the cell membrane, such as steroid hormones, for example, cortisol, estrogen, and amino acid derivatives such as thyroxine. Their mechanism of signaling include ligand traversing the cell membrane and binding with the receptor activating a gene. As it requires transcription of genetic information to mRNA and translation into new proteins, they produce a slower cellular response. So this picture illustrates how a steroid hormone is transported in bloodstream bound to carrier proteins and traverse the cell membrane to go inside the cell to bind with intracellular receptors, both cytoplasmic and nuclear. When bound to its ligand, the receptor ligand complex bind to DNA and induce transcription of genetic information to mRNA and translation into new proteins to produce the intended cellular response. Now that we know what receptors are and how they function, let's discuss about the modulation of receptor activity. There are 4 types of modulators: agonists, partial agonists, antagonists, and inverse agonists. What is an agonist? It is a molecule that can bind with the receptor and trigger a cellular response similar to the chemical messenger, meaning they mimic the chemical messenger action. For example, salbutamol, which is an airway relaxant, mimics catecholamine activity on beta-2 receptors. Morphine is a pain relief medication and it mimics endorphin action by binding to opioid receptors. Next, we have partial agonists. They are molecules that bind the receptor but only trigger a partial cellular response compared to the chemical messenger, unlike full agonists. Therefore, they also mimic chemical messenger activity but with a lesser response. For example, tramadol is a pain relief medication which acts as a partial agonist to opioid receptors. Next, we have antagonists. They compete with the normal messenger for the receptor to bind with the receptor. However, this will not trigger a cellular response because their action is blocking the receptor activity. For example, propranolol, a cardiac drug, blocks the beta-adrenergic receptors. Finally, let's look at inverse agonists. They bind with the receptors but trigger a cellular response opposite to that of binding with an agonist. They reduce the function of receptors in active conformation. Thus, they mimic an opposite action to the normal chemical messenger. For example, benzodiazepines are GABA agonists, which reduce anxiety and fits. Whereas we have beta-carbolin, which is another drug that acts as an inverse agonist to GABA receptors by reducing the effect of GABA receptors, causing anxiety and seizures. So this graph shows the cellular response by these 4 modulators. Agonists increase the cellular response, while antagonists prevent producing any cellular response, and inverse agonists produce an opposite cellular response. Lastly, let's talk about regulation of receptors. They are subjected to regulation by changing the number of receptors and their affinity to the specific messenger. So receptors can be either downregulated or upregulated. The downregulation occurs when a high concentration of the messenger is maintained for a period of time, leading to reduction in the total number of cell membrane receptors. This reduces the target cell responsiveness to the messenger. This occurs by intracellular internalization of the messenger receptor complex by endocytosis, leading to increased degradation of the receptors in the presence of high ligand concentration, and also by desensitization by chemical mediated of receptors in ways that make them less responsive to the messenger. An example is presence of high insulin concentration in the blood leads to downregulation of insulin receptors. Now, this picture shows how TRH receptors are downregulated in the presence of high T4 and T3, which is also termed negative feedback inhibition of thyroid hormone on TSH synthesis. You will learn things like this when you go to endocrine module. Now, this picture depicts how the cell membrane receptors are downregulated by internalization of receptors through endocytosis. Next, upregulation of receptors. This occurs when cells are exposed to a low concentration of messenger for a period of time, leading to increase in the total number of cell membrane receptors. This causes increase in the target cell response and occurs by incorporation of receptors in intracellular vesicles to the cell membrane. For example, uterine oxytocin receptors increase in the third trimester of pregnancy to promote uterine contractions.